Hello, my name is Josh Doherty, and today I'll be presenting to you my PowerPoint presentation on glycolysis, gluconeogenesis, and the role of the enzyme Aldolase gene expression. So in this presentation, I will be going over the glycolysis pathway and the steps in which I'll be going through real quick. Along with that, I'll be talking about the gluconeogenesis pathway and its steps, and explain to you what the enzyme Aldolase is and why it's important for these two pathways. And I'll be talking about the different isoforms in which the Aldolase has, and then finally focus specifically on the expression regulation of the Aldolase B. Okay, so the first thing we'll be talking about is the glycolysis pathway, which is very important to see because this basically focuses on the glucose molecules broken down into pyruvate, specifically two molecules of pyruvate. And a little fun fact here, the glycolysis is the Greek word for sweetened sugar and lysis splitting. So it's sugar splitting. So we're splitting sugar molecules. So it yields two molecules of pyruvate, three carbon compound molecules specifically. So during the sequential reactions of glycolysis, some of the free energy released from the glucose is conserved in the form of ATP and NADH. And by the way, a little fun fact as you see in this little chart here, ATP and NADHs are used in the entire glycolysis process. And we'll talk about the different phases in the next slide. Okay, so first let's talk about the whole process of the glycolysis pathway, which actually has two phases, or two phases, sorry. So the first phase is the preparatory phase, where it prepares the glucose molecule in order to be converted to the pyruvate. And we'll talk more about the payoff phase later. So it's basically the phosphorylation of the glucose and its conversion to the glycerol aldehyde 3-phosphate, which will be used in the payoff phase. So the first step we're going to be focusing on is how this glucose molecule up here is phosphorylated and at the hydroxyl group on the carbon 6 bond. And this actually becomes the glucose 6-phosphate. And by the way, this requires the ATP molecule to be used. It's considered the first priming reaction. So step 2 is where we see the D-glucose 6-phosphate being converted into the fructose 6-phosphate. And one thing I forgot about the glucose being converted to the glucose 6-phosphate is by the hexokinase. So as far as the converting glucose 6-phosphate to the fructose 6-phosphate, this is done by the phosphohexose isomerase. So the third step is where we have the fructose 6-phosphate phosphorylated again. And this occurs at the carbon 1 bond to yield the fructose 1,6-biphosphate. And this is, of course, important in order to create the glycerol aldehyde 3-phosphate, which I'll get into further detail how that's done. And for both the phosphorylations, ATP is required. As you can see, ATP is being used, and I will discuss more about the fourth step. Okay, so the fructose 1,6-biphosphate is split by the aldolase molecule, the aldolase enzyme, I should say, into the two molecules which you see here, the glycerol aldehyde 3-phosphate and the dihydroxyl acetone phosphate. And these are the three carbon molecules needed for the payoff phase, which we'll talk more about later. So this is known as the lysis step, the splitting, the cleaving step that gives the pathway its name. So the dihydroxyl acetone phosphate is isomerized to a second molecule of glycerol aldehyde 3-phosphate, which is why here in this payoff phase, we see 2 molecules of the glyceroldehyde 3-phosphates, which will be used for the payoff phase. So to summarize all what I just said about the preparatory phase, all right, before I go on to the payoff phase. So in the preparatory phase of glycolysis, the energy of ATP is invested, raising the free energy content of the intermediates, and the carbon chains of all the metabolized hexoses are being converted to a common product, the glyceroldehyde 3-phosphate. The energy gain comes in the payoff phase of glycolysis, of course, in which I'll be talking about here right now. So, the important thing about the payoff phase is this is where we have the pyruvates being made. This is known as the payoff, and of course there's more to this payoff than just the pyruvate. So each molecule of the glyceroldehyde 3-phosphates are being oxidized by the 2 molecules. So you have the 2pi, and then more importantly, the electron carrier, the 2NAD+. And by oxidizing these 2 molecules, the glyceroldehyde 3-phosphate, the 2NAD+ becomes the NADH electron carrier molecules. And each molecule, the glyceroldehyde 3-phosphate, is oxidized phosphorylated by inorganic phosphate, not by ATP. And which, by the way, this is where we have the 2pi, is the inorganic phosphate. And this forms 2,1,3-biphosphate glycerates. Alright, so as you can see, the energy is then released as the 2 molecules of 1,3-biphosphoglycerate are converted to the 2 molecules of pyruvate, and that's the whole process of it. And so the 7th step to do this is that the 2 1,3-biphosphoglycerate molecules are being converted to 3-phosphoglycerate, and this is done by having 2 ADPs, which will be converted to 2 ATP molecules. Excuse me now. So the 8th step is where we see then the 3-phosphoglycerate molecules being converted to 2 phosphoglycerate molecules, 2 of them. And so once it actually lets go of the water molecules, 2 of them, they would be further converted to phosphoenol pyruvate molecules. And then finally, those phosphoenol pyruvate molecules will be converted into pyruvate molecules. This is done by the 2 ADP molecules, which are converted to 2 ATP, which, by the way, this actually causes the net yield of 2 ATP. This is how we get our net yield of the molecules used. And the energy conserved in forming the molecule and the forming electron carriers NADH. So the energy is also conserved in the payoff phase and those formations. And of course, that is important for the cell functions. Okay, so now we'll be talking about the gluconeogenesis pathway process, which, a little fun fact there, it's a Greek word meaning the new formation of sugar. So this is done by converting the 2 pyruvate molecules and the related 3 and 4 carbon compounds into glucose. And as you can see here, there's this long process all the way up to it. And I'll be talking more about that. So gluconeogenesis and glycolysis are not identical pathways running in opposite directions. Or of course, it may seem to appear in that image that way. And there's a reason why. They may share several steps. In fact, 7 of the 10 enzymatic reactions of gluconeogenesis are the reverse, like glycolic reactions. With that said, however, there are 3 reactions like glycolysis which are essentially irreversible in vivo and cannot be used in the gluconeogenesis process. And the 3 following steps are the conversion of glucose to glucose 6-phosphate by hexokinase, and you have the phosphorylation of fructose 6-phosphate to fructose 1,6 biphosphate by phosphofructokinase 1, and the conversion of the phosphoenol pyruvate to pyruvate by pyruvate kinase. So, just a little overview about the gluconeogenesis here, real quick, okay? So, in this process, we see here that the pyruvates are being converted to pyruvate carboxylase by the two HCO3 molecules plus 2 ATP. And of course, the ATP is converted to 2 ADP plus the 2 phosphate molecules. This, of course, the pyruvate is being converted to the oxaloacetate, which that molecule is converted by the 2 GTP by the phosphoenol pyruvate carboxylate molecule. I can't really say that at the moment. So this converts into the phosphoenol pyruvate. And of course, this is being converted by the 2 water molecules by the enolase. And this converts it to the 2 phosphoglycerates. And in order for the 2 phosphoglycerate molecules to be converted to 3 phosphoglycerate molecules, the phosphoglycerate mutase converts this molecule. So, with that said, the 3 phosphoglycerate molecules are being converted by the 2 water molecules and the phosphoglycerate kinase into becoming the 1,3-biphosphoglycerate. And of course, there's this step in which the 1,3-biphosphoglycerate is converted by the water and the glyceraldehyde 3-phosphate dehydrogenase to convert it to the glyceraldehyde 3-phosphate. And of course, we have the aldolase, which one thing I will talk about in the aldolase is that it does convert this into the fructose 1,6 biphosphate. Interesting fact here. And of course, the rest actually converts to where the fructose 1,6 biphosphate by water into fructose 6-phosphate. Oh, and I also forgot the fructose 1,6 biphosphate enzyme catalyzes this process to fructose 6-phosphate, and then we have the fructose 6-phosphate by the phosphoglucose isomerase to convert it to glucose 6-phosphate, and then finally we have the glucose 6-phosphate being converted by using water and the glucose 6-phosphatase enzyme to ultimately convert it into the glucose as we know. Alright, so here's an interesting thing about these irreversible steps. While these steps are irreversible, they can be bypassed by special enzymes. And so they have the gluconeogenesis has its own separate enzymes to help bypass these things, catalyzing the reactions that are sufficiently exergonic to be effectively irreversible in the direction of the glucose synthesis. So thus both glycolysis and gluconeogenesis are irreversible processes in cells. So an interesting thing is in animal cells, both pathways occur largely in the cytosol, and necessitating their reciprocal and coordinated regulation. So separate regulation of the 2 pathways is brought about through controls exerted on the enzymatic steps unique to each. Okay, so now that we have talked about the 2 pathways, let's talk about the aldolase involvement in glycolysis. And this is where I'm going to be talking about the enzyme that's called aldolase. So the important thing about aldolase is it's involved in glycolysis' 4th step, which, as you see here, is where you see fructose 1,6 biphosphate divided into these 2 molecules. Remember how we talked about that step 4 in the preparatory phase to where this molecule would be cleaved into these 2? That's because of the aldolase, and this is why it's important. So, as I mentioned earlier, it would be divided into the glycerol aldehyde 3-phosphate, which aka you could call GAP. And then, of course, this one here is like glycerol 3-phosphate, which you could call it G3P or GAP. And here is DHAP, which is dihydroxyacetone phosphate. So this reaction is catalyzed by the fructose 1,6-biphosphate aldolase. And this actually focuses on the fact that it cleaves the fructose 1,6-biphosphate C3C4 bond and to form these two molecules, which is following the intermediate formation within the active site. And that is how the aldolase creates these two molecules, which is needed for the further steps of glycolysis. Okay, so the important thing about this is the fructose 1,6-biphosphate is also involved in the conversion into the fructose 6-phosphate. And so this is due to phosphorylation, and this is the second bypass of through the glycolytic reactions. As you know that these reactions, the glycolytic reactions, are irreversible, so that's why the gluconeogenesis needs those enzymes to bypass through these reactions. So the reactions are irreversible in the intact cells. And so it would need the generation of fructose 6-phosphate from fructose 1,6-biphosphate to be catalyzed by a different enzyme, specifically the magnesium-dependent fructose 1,6-biphosphate. And that promotes the essential irreversible hydrolysis of the C1 phosphate, not the phosphoryl group transferred to ADP. Okay, so let's get a little more detail about the aldolase isoforms here. And they have their own unique expression patterns. And one thing I find interesting about these is that each of these aldolases have a different role depending on the human body. And you can see here that the aldolase A is expressed on the muscle and red blood cells. And aldolase B type, I should just call it, get involved in the functions of liver, kidneys, and small intestines. And then finally you have the aldolase C type, which really plays a role in the central nervous system, specifically on the brains and the nerve tissue. And one interesting thing about these different types is they have a certain percentage of identity, like similarity, and sequence, that is. So we see here that there is a similarity identity between 60% to be exact between human A and human B aldolase. And then you have a higher percentage of identity between human B aldolase and then human C aldolase, which is 68. And then finally the 78%, which is the highest, between the human A aldolase and the human C aldolase. Okay, so all these isosomes, which I have discussed earlier, have strictly conserved residues in the active site, consisting of ASP33, ARG42, Lyse107, and many others. One example of which I want to focus on is the Lyse229, which specifically forms the shift-base intermediate. So each of these aldolase isoforms are unique in their immunochemical distinction, and they have different catalyst properties, such that the ratio of the specific constants for the two hexose substrates of aldolase C is between those of A and B. And aldolases A and B have well-defined roles in glycolysis and gluconeogenesis. As far as the aldolase C, however, there's really not known functions yet, as far as the scientists have studied upon. Okay, so talking about the aldolase B expression here, I will be discussing on the proteins that regulate the aldolase B expression. And 4 of those proteins will be discussed in this entire presentation. But first, I want to focus on the proteins HNF1 and HNF3. So the HNF1 is the first transcription factor involved in the activity of liver-specific promoters to have been well categorized. So it plays a major role in the tissue-specific expressions of numerous genes active in the liver and small intestine. And the HNF1 is synthesized in tissues in endodermal organs, such as the liver, small intestines, the pancreas, and even the kidneys. So HNF3 behaves as a strong inhibitor, counteracting competitively the action of HNF1 and non-competitively the action of DBP, which I'll discuss more later. So both HNF1 and HNF3, okay, could bind to the Aldolase B promoter on overlapping sites. That's why that you have this HNF3 counteracting HNF1s, because both of these could bind to that promoter, the Aldolase B promoter, and that's what's important. So the HNF1/HNF3 binding site is contiguous to an NFY binding site centered on a GCC-AAT motif, and that's a DNA. Yeah. Okay, so continuing on about HNF1 and HNF3, so the proteins serve in controlling Aldolase B gene transcription, and that's why they play a major role in the Aldolase B expression. And HNF1 is very strong transactivator, but not HNF3, since both compete for that overlapping sites in the Aldolase B promoter. So HNF3 counteracts HNF1 dependent transactivation. So interesting fact about HNF3, it also blocks DBP's transactivation ability, which signifies that the transactivation can only be exerted on a HNF1 charge promoter and not on a HNF3 charge promoter. So since the binding sites for DBP and HNF3 are different, HNF3 is a non-competitive inhibitor of DBP-dependent transactivation. Okay, so I'll be talking about the regulation of Aldolase B expression by the two final proteins I'll be talking about is the C/EPB and the DBP proteins. So the DBP is a potent transactivator of the Aldolase B promoter in the heptatoma hepG2 cells, but it's inefficient in the hepatocytes of the primary culture, and while the C/EPP is the other way around, to where it's very active in the hepatocytes, but not in the hepatoma hepG2 cells. So it's a reverse of these two. So in contrast, both transactivators, that is, are active in the albumin promoter in hepG2 cells. Although a poor activator of the Aldolase B promoter in hepG2 cells, the C/EBP is able to bind to its cognate element in these cells, since it partly antagonizes DBP-dependent transactivation, while this antagonism cannot occur through DBP's C/EBP heterodimerization. Okay, so in conclusion, basically we discussed how glycolysis is that pathway which breaks down the glucose molecules into two pyruvates, and we also looked over the process of the gluconeogenesis, which takes that pyruvate and converts it into glucose, and we talked about the aldolase enzyme, which plays the important roles on glycolysis and gluconeogenesis, specifically in the step 4 of the glycolysis. And we discussed on the different isoforms and its expressions, and then we talked about how it was important for the aldolase B expression to be regulated by the 4 proteins, HNF1, HNF3, the DBP, and the what was that? the E/C/EBP. And thank you for watching this presentation. If you have any questions, feel free to ask, and hope you have a good day.